So I did just that. I generated 450 different geographies with different numbers of regions and different values for a handful of environmental features. And then I generated a range of other model parameters to go with those geographies. Next, I simulated 900 different trees with tip states using those geographies and model parameters. After simulating the data sets, I proceeded to apply the multi-fig model that we made. And finally, I compared the output to the input to see how well the model does at recovering the truth. So here I've graphed the results for a bunch of different model parameters. The true values are on the x-axis and the estimates are on the y-axis with 80% HPD intervals. So the better the model does, the closer the points should be to the one-to-one line. We also did a coverage analysis shown in the bottom right of each graph. Those values should be targeting around 80%. So these are those sigma and phi parameters that represent the strength of the effects of different environmental features on each of the core processes. For instance, that one over there in the bottom right, phi d altitude, is a measure of how important differences in altitude are for rates of dispersal between regions. As it turns out, from up there, you can see model does a pretty decent job of recovering information. Wait, I promised lizards. So I decided to use the model on a clade of lizards called Leolamidae, which comprises over 300 species spread throughout South America. These lizards are interesting for a lot of reasons. It's a really diverse group composed of both oviparous and viviparous species, experiencing a wide range of environmental conditions. Many of the species live in these high altitude montane habitats, and there's been some curiosity as to whether these montane environments are contributing to high rates of diversification. So to apply our model, we split South America into those 6 regions, as denoted by a 2019 evolution paper by Damien Eschiere. And in that paper, the authors examined the biogeography of this clade, comparing Andean regions to non-Andean regions and uncovering higher rates of speciation in the Andes. But with the models available at the time, they weren't able to investigate all 6 regions separately or to consider other features of the regions. So I wanted to see whether the multi-fig model would tell us something new about Leolamidae. So I looked at a set of 8 environmental features that I thought might be important for diversification and dispersal in Leolamidae. I looked at region areas, so that's the actual numerical sizes of regions and also a large, small categorization, and whether that would affect rates of speciation and extinction. I looked at region altitudes, so mean altitudes and a highland-lowland classification, and whether they would affect rates of speciation and extinction. I also looked at differences in altitudes between regions, quantitative and categorical, and how that might affect between speciation and dispersal. And I looked at distances between quantums and region adjacency, and how that could impact between region speciation and dispersal. I generated the data using an R package that I'm currently developing called Region Features, which generates summary statistics and categorizations for a set of regions and any number of feature rasters. Then I performed an analysis using the multi-fig model with a fixed tree of Leolamidae. As a side note, we used a neat bit of math called reversible jump MCMC that essentially allows us to choose between models that have certain parameters turned on or off. In this way, we can figure out which of these features do or don't have an effect on a certain process. So our results come in 2 forms. We get posterior distributions that show our estimates for values of strength parameters, and we get reversible jump probabilities that give whether a certain parameter is probably on or probably off. So if 0 falls within the HPD interval for a parameter and the reversible jump probability doesn't show an on value, we probably can't reject the null hypothesis that a parameter doesn't have a relationship with a process. On the other hand, if 0 falls outside the 95% HPD interval and the reversible jump shows an on value, that gives us a reason to think that a parameter may have a relationship with a process, and we can estimate the strength of that relationship based on the posterior mean. We uncovered some interesting results. So here I've got the posterior distributions of some parameters alongside support for a 0 value versus a non-zero value. These parameters, phi w and sigma w for altitude, represent the impact of altitude on within region speciation rates. Our model, surprisingly, didn't find an effect of quantitative or categorical altitude on within region speciation. This tells us something interesting when compared with the results of the Eschiere paper, where Andean regions were found to have higher within region speciation rates than non-Andean regions. One hypothesis as to why our results might be different is the coding of highland and lowland regions. In this analysis, Patagonia is classified as lowland based on mean altitude, but in the Eschieri paper, it was classified as Andean because the south part of the Andes is in Patagonia, which brings up questions. If there really are higher speciation rates in the Andes, is there something else about mountains besides altitude that increases speciation rates? We don't know yet. We did find that one of our features had a significant effect. Specifically, quantitative differences in mean altitude had an effect on dispersal. As one might predict, greater differences in mean altitude contributed to lower rates of dispersal between pairs of regions. Interestingly, categorical differences in altitude did not show an effect on dispersal. That is, two regions sharing or not sharing highland-lowland classification didn't impact dispersal. It's only when we incorporate actual values for mean altitudes that we see an effect. So these results highlight the utility of the multi-fig model for examining the relationships between geographical features and evolutionary and biogeographic processes. We learn something interesting when we're able to examine the potential effects of many environmental features, both categorical and quantitative. And with its general framework, the multi-fig model could be applied to many different study systems with different taxa, regions, and geographical features. I'm currently working on a manuscript about the multi-fig model, the simulation study we did and how we applied it to Leo Lamidae. I'm also planning to publish that region features R package that I created to generate the data matrices from feature rasters. And in the future, we are working to extend the model to allow for the inclusion of things like paleogeographic information, as well as jointly estimating phylogenetic trees. I'd like to acknowledge all the members of the Landis Lab who've given feedback on this work and thank the LEC for the opportunity to give this talk.
We'll have a quick time for a few questions if there are any.
For the Patagonia thing, could you recode the geographical distributions so that they were correctly assigned to the Andes or not?
Yeah, absolutely. So the next analysis actually that I plan to do is to replace that in this particular model and see if we find the same thing that happened in the Eschieri paper or not, which would be an interesting finding either way. Yeah, that is next on the agenda.
How complete of a phylogeny do you think that you need to do these types of analysis that you do?
Don't know. I did not do any like simulations with removing taxa or anything to have a look at
it. Wonderful talk, Sarah. How long does your RGM-CMC take? Was this like for how many 350 animals? Was this pretty quick or?
For the empirical analyses, yeah, it was like 14-ish hours. So not too bad. Yeah. Awesome.
Great. Thanks so much. Up next, we have Trinity from the Strassman-Queller Research Group, arriving from the Indian Institute of Science, Education, and Research in 2017. Trinity largely studies evolution of the dietary niche breath and social amoeba. To your right, let me just switch to the. I'm handing you that.
Hello, is this Michael?
Should be. Sorry, one sec. There we go. There we go. And then, yeah, let me just switch it for the Zoom as well. Sorry. Okay, yeah. So, all right. Let's take it away.
Is this on?
No, yeah, that is, you have that.
Yeah. Okay. So organisms vary widely in their diet. Some are diet specialists such as the koalas over here that feed majorly on eucalyptus leaves, whereas others, such as the coyotes, are diet generalists that feed on very many prey species. Now, specialists have honed their ability to process and catch specific organism or prey, but diet generalists must deal with organisms that vary their nutritional quality or their defensive mechanisms. So how do generalist predators handle diverse prey? Now, in a large carnivore such as a lion, it's like some combination of speed and strength, and it's just chasing this animal and killing and is able to eat it. But an in-depth analysis into the mechanisms of maintaining a broad diet breadth or the challenges associated with it is impossible in lions. So I study them in the next best thing, which is now most of you in this room know Dictyostelium discoideum. It's a social amoeba and it's famous for its multicellular cycle. But today I would like to reintroduce this as a super generalist predator of bacteria. So Dicty is found in cool forest soils and it feeds on bacteria when it's a unicellular amoeba. And like other protists, it can feed on many bacterial strains. So Devi from our lab and team isolated 174 bacterial strains that are found in close association with Dicty in the wild. And 77% of these strains are edible. And these strains span 4 bacterial phyla. This is some of my own data looking at the growth rate of Dicty on 23 different soil bacteria. And basically, you see there is a nice variation in the doubling time of amoebas. They grow really well on some and not so great on others. So how does Dicty handle diverse prey? So what do I mean by that? I mean, are they a fixed generalist? Do they use one common mechanism or a tool to hunt all prey species? So I refer to this as the hammer hypothesis. So it's like Dicty has this one tool, hammer, and regardless of the differences in the prey's defensive mechanisms, it is the same tool to hunt them all. Or are they a plastic generalist? Do they have different combinations of tools to hunt different prey? And I refer to this as the army knife hypothesis. Basically has a few different tools, uses different combinations to hunt different prey. So I address this question by performing two simple assays. So the first one is called as switching cost, where I look at if amoebas experience a reduction in growth rate when they are switched to a new bacterium compared to controls that do not experience a switch. So that's switching costs. And another experiment is called concurrent cost, where I look at if amoebas experience a reduction in growth rate on multi-prey communities compared to expectations from their single prey communities. So let's take a look at switching costs. So basically, over there you have amoebas that are conditioned to bacterium A. They are plated with bacterium A, and those are the controls for the experiment. And the switches are amoebas that are conditioned to bacterium B, and then plated with bacterium A. So these guys are experiencing a switch. And now we want to see how many amoebas are there after 3 hours from setup in this experiment. Okay, so I did this experiment on 5 Dicty strains, and I did this on 3 pairs of bacteria with 2 reciprocal switches, so which means I looked at 6 different switches in total. Okay, so basically, yes, switches perform poorly compared to controls. So this is just the main effect. The amoeba numbers on the y-axis, and the light red is the controls, and you see there are more amoebas in the controls than switches. And just taking a closer look at all those different 6 different switches, generally we do see that the controls perform better than switches. Now, like I mentioned, so I'm looking at these switching costs like very early after I set up this experiment. It's like within 3 hours from setup. I was curious to see what happens to these switching costs when I let this experiment run for a longer time. And I did this time series on only one of these pairs over here, Agrobacterium and Pseudomonas. And basically, okay, so this one is just showing early growth rate again, where I see that the controls are doing better than switches. But then if I look at late growth rate calculated between 24 and 27 hours from setup, the controls and switches are similar now. So like the switches have caught up in their growth rate to controls. So basically, yes, amoebas experience a reduction in growth rate when they are switched to a new bacterium, but these switching costs are temporary and amoebas eventually catch up in their growth rate. Now, of course, amoebas are more likely to encounter like many species of bacteria at once, and it's not like they feed on one thing the entire time and then move on to the next. So I looked at if amoebas experience a reduction in growth rate in multi-prey communities. So for this experiment, I had like 5 single species communities listed over there, and then 5 3-species communities that are a combination of these single species listed over there, and then 1 5-species community. Okay. So the first thing to do is estimate amoeba numbers on these single prey communities, and I have the data over there. And then to calculate the expected amoeba numbers on these multi-prey communities. So for example, if I wanted to know what's my expected amoeba number in a prey community that has AT, SM, and CT, it's just an average of my observed amoeba numbers in those single prey communities. But then now I will compare these expected numbers to what I actually got. Okay, so this is the first one, AT, SM, CT. So the light green is the expected number and the dark green bars are the observed numbers. And okay, yeah, so for this one, there is a concurrent cause, so they do perform worse than we expect. Similarly, for this mix of prey, also for this one, but I mean, the happy story, I mean, it has to become sad at some point. So basically, it's not true for this SMCT PK mix. It seems like they do just as well as you would expect. And in this other case, they even do better than expected. But then in this 5-species community, they are performing worse again. So going back to this question, how does Dicty handle diverse prey? Are they a fixed generalist or a plastic generalist? So the results from my experiments support the idea that they are a plastic generalist and they use different combination of tools to hunt different prey. So the logic behind that is basically, okay, so if Dicty uses the army knife technique to hunt different prey, so when it is switched from one prey to another, it has to switch between its tools. So it experiences a temporary reduction in growth rates when it does that. But then eventually when it is used to eating the same bacteria, it's fine. So it's a temporary reduction in growth rate. So that's why you'd expect to see switching costs. And then once again, if it was an army knife and it has to deal with many different prey species at once, it's unable to deploy its multiple tools at once. So it's just performing worse than you would expect. Yeah, so yeah, that's the takeaway. So Dicty is basically a plastic generalist. It has different, at least has a few different combinations of tools on different prey. My future plans are to experimentally evolve these Dicty on a monophagous diet and see what other fitness trade-offs or behavioral consequences of this are. Yeah, that's my talk. I'd like to acknowledge the Queller Strathman Lab, especially Joan and Dave, and Debbie for help with some of the switching cost experiment, and Rachel, my undergraduate mentee, for help with the concurrent cost experiment.
We have a little bit of time for questions. Awesome.
All right, that was a fabulous talk. And I agree that Dicty is really the second best after Lyme. So my question is, in the longer term assay that went out to 24 or 27 hours, do they still have plenty of food left, or are they reaching like an asymptote as they become resource limited?
Definitely after 27 hours, at some point they are going to start aggregating, but I didn't observe any aggregates. So what Dicty does is when it runs out of bacteria, when enter the multicellular cycle. So I didn't see that on those plates, which means there is still plenty of bacteria to eat on the plates. Yeah.
Hey, great talk. So you showed at the beginning that the amoeba have different growth rates on the different bacteria, which might reflect that they would prefer certain prey types over others. Do you know, or is it possible to tell when you present the amoeba with 3 or 5 different bacteria species, are they consuming all of them equally, or are they eating more of certain species than others, and how might that impact what you're seeing?
So in my experiment, I do not test that, but just hoping that because of those prey densities, they at least encounter these different prey. But Callum is going to vary prey densities and look at whether they prefer the more common prey in this mix. So maybe next time he's going to present a talk on that. Yeah.
Great talk, Trinity. Have you looked at whether the costs of prey switching are associated with phylogenetic relatedness of the prey?
So I do plan to do that, but then on just these 6 switches, it's not enough data to do an analysis like that. Yeah, but hopefully I can do a few more switching cost experiment and see what that looks like. Yeah.
Great talk.
Thank you.
Cool. Next, we have Wen from Ken Olson's lab, 5th year PhD student. I'll get your talk going. Sorry. Oh yeah, you get to see my desktop, sorry. That's what all the whispering was, yeah. Great. Yeah, I get that. 1 sec, sorry. See what happened. Right.
And this one happens. This.
I think it's not. So let's see. Try.
Okay, thank you. So I'm Wen from.
Actually, yeah, try it the other way.
Okay, try again. So I'm Wen from the Olson lab. Today I'm going to talk about copy number variations and local adaptation in white clover. So what is copy number variation? Copy number variation is a kind of genomic replication with the base unit bigger than 1,000 base pair. It is pretty common than we thought. It can contribute up to 10% of human genome, and sometimes it can be functional. For example, in a soybean, you can see the green color means the base unit over 31 kbp replication unit. And in the William 82 cultivar, it only has 1 copy, but in the 38 cultivar, there has 10 copies. And scientists have found this high copy number of these replications is related to nematode resistance. And this knowledge has been applied in multiple commercial soybean cultivars. And let's talk about how the copy number variation related to clover. Clover has a spatial defense mechanism called cyanogenesis. When an herbivore approaches the clover, it can rupture the cell and make the substrate and enzyme in the cell contact each other. The substrate is a cyanide precursor, and the enzyme can break the precursor and release the cyanide. And cyanide can make the herbivore sick or dead. And this process pathway called cyanogenesis. And biochemically, the precursor is synthesized by 3 genes. They are physically linked, and we call this linked gene cluster, AC gene cluster. And the enzyme is encoded by the LI genes. And the Olson lab has found there is polymorphism in these two genes, and the polymorphism is a presence-absence polymorphism. The absence means whole gene deletion. So a clover plant need to have both the AC gene cluster and the LI gene to be cyanogenic, that is, can successfully produce cyanide. And so phenotypically, there are 4 combinations, which is AC plus, LI plus, AC minus, LI plus, AC plus, LI minus, and AC minus, LI minus. And only the AC plus, LI plus combination can produce cyanide. We call it cyanogenic. The Olsen lab also find the allele frequency of this AC gene and LI gene is related to minimum winter temperature, with higher frequency of this allele in a warmer climate, which indicate the polymorphism of AC and LI gene is likely to be a result of local adaptation. Our story does not end here. So 15 years ago, the young Dr. Olsen, he did a cross between 2 cyanogenic clover strands, and it gives rise to several F1 offspring. And he also sequenced the AC gene cluster in the offspring, and he find some individual may have up to 3 alleles, which doesn't make sense. And the best inference of the parental genotype could be something like this one, but even the best inference fail to explain the result, because if one parent is homozygote for the AC gene, all the offspring should be like a AC plus phenotype. So in order to resolve this problem, the young Dr. Olsen made a bold move to make the copy number variation hypothesis. If we move the allele into the same place, so like AC1 plus AC2, and the other allele is a deletion, and then by this model, we can explain the result well. And this is a kind of copy number variation. However, this hypothesis has not been tested until we finally sequence the clover genome. Clover is a tetraploid species with 32 chromosomes in 2 subgenomes. So it's not easy to sequence. But with the long sequencing technology, we finally get the genome data. So you can see the left panel, there is 2 subgenomes, and the color links show the gene synchrony between the 2 subgenomes. So the copy number variation should stand out if we align 2 homologous chromosomes, like here. And now I'm going to zoom in the chromosome 2, where the AC gene cluster locates. So please look at the left panel, and the x-axis is one of the AC allele, and the y-axis is the other AC allele. And you can see there are 4 genes in the AC cluster in the AC1 allele with 1 gene duplicated. And each vertical line has 3 hits in the AC2 alleles, which means there are only 1 gene copy on the AC1 allele, but there are 3 copies in AC2 allele. In the right panel, I align an AC plus allele to an AC minus allele. And you can see here is a huge gap like in the AC minus allele, which is consistent to the presence absence mutation. And this plot can better visualize the alignment. You can see each gene in the AC1 allele has 3 hits in the AC2 alleles. And we also find this replication pattern is complicated. If we define 2 replication base unit on the AC1 allele, it has 3 hits on the AC2 alleles, but they are overlapping. we did find some similar result for the AC genes, but due to the limited time, I'm going to cover it today. And we believe there are more variations in the wild. Then we want to see if this copy number variation functional. So here, the x-axis is the copy number of the AC gene cluster and the LI genes. The y-axis is the cyanide production level. So yes, we do find there is a strong positive correlation between the genes in AC gene cluster to the cyanide production level, but not for the LI gene. Then we want to see, is this functional copy number variation a result of local adaptation? To do this, we sample 43 population, including 415 individuals across North America. And this is the map with the color shows the minimum winter temperature. And we did correlation. Yes, so like please see the left panel, the y-axis is the cyanogenic polymorphism in the presence and absence form. And consistent to the previous study, we find a positive correlation. And the middle panel, the y-axis is the copy number. So yeah, we do see there is a strong correlation between minimum winter temperature to the copy number of AC and LI genes. So yeah, the copy number variation is likely to be a result of local adaptation. So what's next? We want to see a more comprehensive result. First, we want to see if we can see the AC and LI copy number variation signal from the genomic scope. In this plot, we try to use minimum winter temperature to explain all genomic variations. And here are the AC and LI genes. You can see they do stand out as a signal, but they are something much more stronger than that. So my next step is to find what are those significant strong peaks, how and why they are related to the environment. So I need to thank you, my advisor, Ken, and also several people in my founding souls, and it is LEC for giving me the opportunity to have a talk here today. Thank you.
We have a few minutes for questions. All right.
Great talk, Wen. Do you know any other examples in plants or animals where copy number variation varies with temperature?
Not the temperature, but there are several copy number variation in human, and I know there's a copy number variation for amylase. So for the human population with rice for their main diet, they usually have more copy number of the alpha amylase, but I'm not sure about temperature. Oh, I know one. It's so for the people living at very nose place, like Alaska, they have a copy number variation for fatty acid metabolism. And it could be a male adaptation if those people live in a warm climate, but it may have some benefit for them living in that very cold climate, and their dietary is mainly on a pretty fat meat or something. Yeah.
That was a great talk. I have a related question. Is it that you think that this coldest temperature of the year is directly acting as a selective agent on this allele frequency, or do you think that it's something indirect like herbivory, like whether you have winter, large amounts of plant matter for foraging in the coldest months?
I personally think it's not direct. So my speculation is the minimum winter temperature can cause some other environmental factor, and that can then related to the AC and LI gene frequency. And herbivore pressure is a well-known hypothesis, and several people try to test this hypothesis, and sometimes it show positive result, but it's still controversial, which is the real factor driving this adaptation. Yeah.
Any other questions? Okay,
There's one from Justin.
thank you, sir. Listen to that.
Thank you. Do you expect to see similar pathways of changes in copy number across alternative temperature gradients? For example, if you found clover in South America, the southern cone of South America, would you expect to see similar variations, or are there alternative genetic pathways to acquiring the same phenotype as well?
I don't know, because like previous study, they quantify this AC and LI in binary way, and they did find this correlation occur in multiple places around the world, like in Europe, in Australia, and so I believe it is pretty likely to show a similar pattern. I would say that need to be validated.
Awesome, thanks so much, Wayne. All right, up next we have fourth year PhD student in Jonathan Moss's lab, Jhan Salazar, who is an expert in thermal physiology, and particularly in anoles. All right, and let's cue up your thing. Great.
Good afternoon, everyone.
All right, sorry, 1 sec. Sorry for the delay. There we go. Wait. Yeah, sure.
All right.
Well, I need to put to the thing. All right.
Hello, everybody. Today, I'm going to talk about something that I did last summer in Colombia. Basically, I was doing like I did a field trip last year in Colombia for 2 months to do some thermal physiology stuff. So I'm going to talk about that today. First, I want to thanks to all the institutions and people that helped me in those 2 months. Without them, any of this would have been possible, because we spent a lot of days and a lot of nights just looking for lizards. So before we start, I'm gonna kind of give you the outline for my talk. First, I'm gonna show you why I do for research. I mean, I'm in Jonathan's lab, so you know that I work with lizards, but I'm gonna be more specific about what I do. Then I'm going to kind of talk about the thermal physiology in the Colombian Andes and where is my project moving from here. Okay, what I do for science, basically, obviously I work with anoles, and I try to use ecology, natural history, thermal physiology, and phylogenetics and genomics to understand patterns of evolution of an adaptation to basically in the Andes and to mainlands and islands. But today I'm just gonna focus on this small part of my research. Okay. First, as we know, evolution occurs unevenly. So this means that some characters, some features evolve faster than others. And basically, this is kind of an example of that. This is the hemipenises and the albinus lizards. That in this research said that the hemipenises evolve way faster compared to any other trait of their morphology. So this means that there are some factors that make that evolution occur slow, but there are others that make evolution occur fast. So, and this is how my question of research goes. That is like how elevation may affect the evolution of thermal physiology. And how I did this, again, I went to the field for 2 months and I did a bunch of thermophysiology experiments. And well, how do we do this? Basically, we use the performance curve, and we can start different type of variables here. And we have a body temperature, and we have performance. Performance can be sprint speed, respiration rate, and any other trait that measures performance. And from here, we have just I measured two of the different traits that are critical thermal minimum and maximum, which are basically the minimum and the maximum temperature that organism can have before they lose their motor ability. We have in the cold spectrum, we have cold tolerance or CT max, or CT min, and in the warm side we have heat tolerance or CT max. So again, I said I work with anodes, and are distributed like in the neotropics. They can be found in islands and mainlands, and they go as low as the sea level and as high as 3,700 meters above sea level. And just kind of to show you some of the lizards that I work with when I was in Colombia, I can just show you some of the examples. And just so you know, these are my favorite species right here. That's why there are 3 pictures of that one. Okay, I don't know how many of you know, but in Colombia, the Andes is divided into 3 different independent mountain ranges. We have the Western Cordillera, the Central, and the Eastern Cordillera. And all of my research was done in the Western Cordillera, which is the one that it rains the most. So basically, with those 2 months, it almost rained every single day while we're in the field. Almost not. So, and again, I went to different sites along the elevation. So I started like in the lowest side of that Western Cordillera, which was like basically almost at sea level. And we went up in those different sites to almost 2,500 meters. So it was a lot of walking, just so you know. So, where did I go? I went to 2 national parks. I went to an eco lodge and a black community. So if you want to talk about this, you can ask me later why I just emphasize this, like it's different from the rest of the sites I went. And why I wanted to go to Colombia. Basically, in the mainland, we have the most diversity of animals. And in Colombia is the most species country of animals. We have 80 species. We had 78 2 weeks ago, but now we have 80. And also in the mainland, and those are way more hard to find compared to the Caribbean. They have low densities, and they are super hard to see. Basically, as you saw, they are almost green, and the forest is green. So, when we went to Colombia, we took a bunch of data. We took obviously the thermophysiology experiments, but we also took a lot of other data that some students in Colombia are using for doing their undergrad thesis. And obviously, because some of the students in Colombia doesn't have the opportunity to go to field trips, I took many of them to show them how to collect tissues to do DNA extractions and how to prepare the specimens for museum collections. And something really cool is this picture. I love this picture. She was my advisor during my undergrad thesis. And this is Camilo, and I was his advisor during his undergrad thesis. So it was really cool to have them two working side by side, just doing some tissue extractions. So, but I don't know how many of you have gone to the field, but not everything goes as planned. So I have been here in San Luis for 4 years. So I have gone to Dominican Republic, Puerto Rico, and Miami to look for animals during the day. And I said last year, okay, I have all this experience now. In Colombia, I usually look for animals during the night because they are easier to find during the night. So last year I went okay, let's do it during the day. I have all this expertise now. I found only 2 during the day. So this is those pictures of those ones. I don't think we're gonna find any. So just wait until then. I just go look for them. And as I said, we went during the rainy season, so there were a lot of days that we just stayed in the station because it was raining and there was no way that we couldn't go outside and look for the lizards. And sometimes we went outside and we just got rained out. And we rented a car. We broke it 3 times during 1 week. Okay, let's now jump into research. I have 2 hypotheses to what I did. Again, I wanted to see how thermophysiology changes along the elevation. And for CT I was thinking the hypothesis was that it was going to change as you go up in elevation, while CT max, it was going to remain stable. And this is within species. And between species, basically, we thought we were going to find the same pattern. And why is this? Basically, as you know, lizards move around. So there is this hypothesis called Boyer effect that basically states that thermal regulation acts as a driver of evolution. So, and why is this? Basically, you know, in the mountains, depending on where the node is, it can move in a different way. In the mountain top, since it's very cold, it tends to bask and take more sun just to get warmer. While in the lowlands, it usually go to the shade to be warmer. And this fact that they're moving around might change how the city man or heat tolerance might change. That's why I was expecting to be stable as you go up in elevation. So for our first hypothesis, I have this beautiful and not super tiny, like this big, called Anolis Anthony. And for city mean, we found what we expected to find. You know, city mean goes decreases as we go up in elevation, but for city max, we found basically the same thing. So we weren't expecting that. So and then I can say, oh, like maybe it's for this species only. Let's see what happened when I combine all the species that I found. And I found, I think, like 17 different species. And again, we have the same thing. We went there and okay, city mean is decreasing as we go up in elevation. What about city max? The same happened. I was like, oh, I have no idea why D6. So, and then I thought, okay, maybe they're like this decreasing as we go up in elevation, both of them. What about how fast those two are evolving? Since they're following the same pattern, are they evolving at the same rates? I thought, yeah, that might be it, but no. City min is evolving way faster, 3 times faster than city max is. And in the islands, also they have found this same pattern, but they say that city max is evolving less faster because they can move around. They can just change how their thermophysiology evolution goes. But in the mainland, no one actually knows if or how animals behave. So you don't know if they actually go to bask in the highlands or they just hide in the shade in the mainlands. So basically, the results for today, again, this is an ongoing project. I have a lot of analysis to do, but basically we found a really cool pattern that both city min and city max are changing as we go up in elevation in both within species and between species. All right, kind of a summary. Again, let's show you the same slide I showed you before. All of these things are going to be determined or going to work with them in some amount during my thesis. But we found that thermal physiology is changing between and among species. They basically follow the same pattern that city min and city max are decreasing as you go up in elevation, and that those two physiological traits are evolving at a different rate, and that obviously animals have evolved and try to live in the mountain tops, and again, like basically the same thing. So, but what I want to do for now on basically is I want to know how mainland animals are behaving. Are they doing the same thing that they are doing in the mainlands, or they are just doing different stuff? Second, I want to see if this same pattern I can find the same pattern in the central and eastern Cordillera of Colombia, or it's only a western Cordillera phenomenon. And the third one is I want to see how or if any which roles genes might play in the thermophysiology evolution of Andean animals. And yeah, that's basically what I had for today. Thank you.
We have a few minutes for questions. Awesome.
Awesome job, John. I'm curious, across the elevation, do you see like differences in like the average body size of the different species you're looking at? For example, like are there mostly smaller bodied species up at higher elevations or lower elevations, or is it pretty similar?
They're like smaller species in the lowlands and bigger species and in the highlands, basically the same thing.
That was a great talk. I guess I didn't get how you measured the evolutionary rates of the CT max and
Basically, there's this I forget, like method, it's called Adams method. You basically kind of add each trait, like CT min and CT max and per each species, and then just basically run the code. And it tells you how different they are. So they is give you obviously the minimum, like the evolutionary rates, and it give you a confidence interval, and basically that's it. And it's a really, I didn't know how to do that until 3 weeks ago. So
Great work. 2 questions. Did you design the T-shirt and
yeah,
give them out to everybody?
Oh yeah,
How do I get one?
we have to go to Colombia with
Oh.
get the new version, the 2023 version.
Second question. So what is your strategy for finding anoles at night? Are they just like sitting out and they're easy?
Oh yeah, you can just, they're just like literally in like a leaf and fern leaves just there, like, because it's really cold, they just don't move. So just go and just grab them, just put it in the bag.
So if they need temperature, you know, what are they doing during the day? If they need warmer temperatures to be active during the day, are they like higher up in the canopy or?
We don't know, because they're like extremely hard to find. We don't really know where they are because you just don't see them. Yeah.
So you can't tell from the limbs and the toes whether they're trunk or ground? No?
Yeah, you can tell that. But again, they're just hard. Even we know that we also have the ecomorphs in the mainland. We have like 3 more, I think. And you might expect that, you know, based on that where they're gonna be, but they're not there. I don't know, they may just hide during the day and just like come out during the day. I don't know. When that doesn't happen, just they are like diurnal species. Any other question? Yes, Shane, sure. Justin.
Oh, yeah, thanks. One question, is CT min related to CT max when using across species data?
Not really, no. But I mean, for at least one, I just have. That's how we measure. Just for this species, like kind of the only one. This is a really weird, strange species that is the one that have like the coldest and the warmest CT max. They call the CT min and the warmest CT max, but that's basically like the only one that those two are kind of related. But again, this is a really weird and unique species. So, but like for the rest of them, they are not related. They just no.
I think we're just about out of time for questions right now. However, if anyone would like to ask any speakers anything else, we will be having yeah, so we'll have a reception at across stock 322. Bring with interest, there will be food and drinks available. Thank you all very much for coming.